Hello, my name is Alexandre Chicolet. I'm a postdoc at Sorbonne University, and today I'm going to walk you through the virtual lab called Plankton Genomics, which is available in Blue-Cloud, and more specifically through the use of a notebook called Projecting Plankton Functional Gene Clusters, which you will find useful to address the hackathon topic, Understanding the Ocean, especially challenge 1B, how might we tap on available environmental and molecular data to predict changes in ecosystem services provided by the Atlantic Ocean microbiome. This notebook will provide you with habitat modeling and mapping tools. So in this video, I will show you how to use the R code and underlying pipeline of this notebook, which you can find. So let me share my screen. So here we are in the Blue-Cloud entry page. On the right, you will have demonstrator, Plankton Genomic. You click on demonstrator 2. And on this new page, you will find on the left a shortcut to notebook 2, which should look like this with these 4 files. Here you will find a Readme file that will explain you the installation procedure, the handbook, if you prefer having it in Word document, so with all the technical documentations, and a zip file where you will find all the codes as well as the technical documentation with the included codes. So very fast for the Readme file, you will again find the links to the zip file where you can have all what's necessary for the notebook. These zip files should be downloaded and copied into a Jupyter instance, or if you prefer, locally. So once you are in the Jupyter instance, which looks like this, you can have your zip file, unzip it by opening here a command prompt and copy the codes that is in the Readme file. So the unzip, and also when you connect to Jupyter Hub, it might be that all the libraries or packages are not installed for whatever reason. If it is the case, just copy this code and this one, respectively for R and Python, into the command prompt, and you will have all the package installed it will take roughly 1 hour. So now concerning the files that you have once you unzipped all this, you will have service 1 IPY notebook. So this is all the technical documentation with the code in it and several folders. So code folder that will contain all the R scripts, function that will be for custom function that are called by the R scripts, data, which is a temporary folder for temporary data file that are passed through the modeling procedure. And then you have data space, which is a permanent repository where you will find Plankton genomic datasets, the features, so the environmental data for this notebook, which are one of both data sources. Speaking of data sources, so you have in the features different layers of environmental data that are climatologies, where we calculated the mean, the standard deviation, median, etc. over the 2005-2017 periods, and they are accessible here. The other data source are genomic data, which are stored in a Postgres database for computing reasons. So the data are quite heavy. So the first step, I will explain that a bit more detailed later, of the modeling pipeline will be to query this database to get the genomic data we want. So once we have all this set up, we can now speak a little bit about the code itself. So here you have some reminders of the structure, and we will first start by loading all the necessary functions. So first, you can use this code to define the Blue-Cloud directory, which is this one. You can verify it by opening an R console and do a get WD to be sure that this is the right directory. And the data directory that corresponds to the feature data that are in the data space. These ones should not move. Once we did this, you can load the config file, which will basically load in your environment all the necessary package libraries and so on, as well as some permanent parameters for the model. I do not recommend moving these parameters unless you're an expert in machine learning and gradient boosting. I will come back to that later. Once the config file is done, you can load these 4 functions, each of which corresponds to one step of the modeling procedure. So first we'll query the data, then parameter the model, evaluate it, and when we are happy, do the Bootstrap predictions. So all the model has already been run in this example. It takes about 1 hour to run, so I spare you the waiting time in this video and have already everything prepared. So now we enter in the model. Step 1 is called through the query data function, where you can find the general principle here and input and output description. This is the case for all the functions that I will present you. So this data takes the Blue-Cloud pathway as input, the KEG. So KEG-P is for KEG pathway. So all the genes in our entry data are annotated, if it's possible, with the KEG nomenclatura. So this corresponds to, you have the link here, to the KEG pathway database, which looks like this. And the KEG pathway database looks like this, where you have a list of all the metabolic or other type of pathways that you can find for genomic data. Here we use as an example 00190, which corresponds to oxidative phosphorylation, so all what's related to respiration with Plankton. If you click on it, you can have some additional information about this pathway and all the modules, etc., that are within this metabolic pathway. So here we select the pathway that we want to model. Because there is a lot of genes that were clusterized by similarity of sequences, and what we will model are not the genes, but the clusters of these genes. And here you have a list that selects clusters that are at minima present at 80 Tara ocean station, so that they have a certain spatial coverage. Genes that contain gene clusters that contain at least 5 clusters, so avoiding single or 2 gene clusters that might not really be clusters, and a maximum here 25, but we should avoid clusters where you can find all type of genes and that may not have a significant ecological meaning. When querying the data, you select also the corresponding environmental metrics at each stations where you have the presence of these clusters. So here we will select the climatology data that are calculated with the mean, the standard deviation, as well as the dist and bathymetry parameters. And finally, a relative argument. So the data are the reads of genes per stations. So the reads of clusters here per Tara Ocean stations. The reads can be seen as a proxy to abundance, and the relative will do the conversion to relative abundance if you wish. So it's a true-false argument. Once you run this, you will have different messages in the console, just telling you that the query is happening. And at the end, the number of stations, number of environmental features, and number of gene cluster targets are printed. Once you're happy with the dataset, you can go to the step 2, which is training the model. So to train the model, we use a Python library that is called MBTR for multivariate boosted regression tree. You can find the link here with all the documentation of this function and library that is used. So basically, this library will perform multivariate gradient boosting, so several targets, so several cluster model at once. So basically, we will have one model for all the clusters that we've selected, composing one metabolic pathway. And this will read the X features and Y targets data that were printed by the query. This model run function calls the step 2. So once again, we have the Blue-Cloud directory that is given in the function. Hyperparameter, which is a data frame with different combinations of modeling parameters. So learning rate, so the rate at which the model will learn from each consecutive trees that it will build. So that's gradient boosting specific parameters. NQ, which is the number of quantiles the model will test for each split in the trees. And mean leaf, which is the minimum number of data in each terminal leaf of the tree. So that will control the depth of the model. The model is run with cross validation, so n-fold cross validation. Usually we do 5-fold, which is quite adapted for roughly 100 data. And so 1-fold is kept for evaluation only and will be used in step 3. So I will come back to that later. The model takes roughly 10 minutes for 1,000 boosting rounds. So once you run this, you will need to wait a little bit until it's completed. Once it's completed, you will have printed the loss function, which here is mean square error with the number of boosting rounds and also the selected best hyperparameters. So the one that produces the minimum loss among all the cross-validation and folds. So the mean, average, minimum, and the number of boosting round corresponding. Here, it's the red one, so hyperparameter number 1 that has been selected. Concerning the outputs, it will just print a file with the model object in it, but you should not interact with the outputs of this function. Once the model is run, we can go to the evaluation. So we will load again the model automatically on the best selected model, and the evaluation function called through model eval takes once again the Blue-Cloud directory. If you want an evaluation by targets or a multivariate evaluation, and also if you want to print or not the variable importance. Here we choose not to do it because it's quite time consuming, because the model will look for every loss gain for each boosting round, each tree, etc. So it's quite time consuming. Once you do this function, you will have printed the multidimensional R-squared and the root mean square error, which both are here in this example quite average or good given the number of data, the number of targets, etc. that we gave to the model. And you will have a series of graphs for each target. And here CC corresponds to connected components, so the cluster number, station number, the relative abundance, and let me show you maybe an example that is a bit more visual. Here, for example, so in black you have the real data, and in gray you have the model prediction. So we see here that the model is quite following the data, maybe not as much in high values, but we have the overall shape. And so you can have the difference per station and see which station here, for example, in the Southern Atlantic Ocean, that are not very, very well modeled for very high abundance values. The variable importance, if you choose to run it, will be printed here. So it will just be a bar plot with the variable names and the relative importance of each variable. So once we evaluated the model and we are happy with the fitting quality of the model, we can go to the last step, which is projecting the functional gene clusters via Bootstrap. So this is called through the proj function. Once again, same parameters, the directories, and here you should put the same environmental metric selection than in the model training. So this will retrain a new model and bootstrap new models on all the data with the same number of boosting rounds and same parameters as the selected model. It will give you an output that is raster of all the targets. So the projection, the spatial projection of all targets given the environmental value at each pixel. So this will take more or less as much time as the model run function. So roughly 10 minutes for 1,000 boosting rounds. And then you have a series of function and objects that are printed so as output of the model proj and can be accessed through graphical functions. So once you run the projection, you will have a function to plot the legends of your plots. So here we choose to put the relative abundance as well as the standard deviation on the same color scale. So the color will control the abundance and the saturation of the color will represent the standard deviation. So you can plot this with the legend plot function and give it the proj call matrix, cutx, and cuty functions, objects. Once you did that, you can plot the maps of your functional gene clusters with the map proj function. So again, you give it the proj object, call for color, and the target ID, which can be all the target at once or one target, if you do not wish to plot all the targets at once. So you will have one plot per target with, for example, here for cluster number 5, we can see that the abundance around the tropic zone is quite higher, especially in the Pacific than in the Atlantic. The standard deviation doesn't seem very high because you have very bright colors. Here for this one, we have very low abundance, for example. And same again for example, cluster 18, which is more polar abundant in the polar region. So these polar regions have more potential of having this gene cluster. Now that we have maps, we don't really know to what they correspond. Well, they correspond to clusters, but we can access an object within the query object. So you can open a console in the PY notebook and we can check for a query connecting component description, which is the table with all the cluster numbers and the corresponding functional description. So the two I showed you, for example, number 5, which is related to pyrophosphatase, and number 18, which is also related to pyrophosphatase, for example. So these one, all the abundant clusters here were related to pyrophosphatase, which is, for example, interesting because you don't have the same cluster coding for the same function. So different genes, a diversity of gene depending on where you're situated in the globe, coding for the same thing. If you want more plots, you can also have the possibility of doing a correlation plot between the targets. So here we can see that cluster 5, cluster 11, and cluster 18 are quite different of the others in terms of spatial correlation. So this is a point-by-point Pearson pairwise correlation. And yeah, I hope that this video has helped you in a bit understanding how to use the notebook projecting Plankton functional gene clusters. And you can find all the documentations in this PY notebook file. So detailed description of all the clusters, the outputs, inputs of the functions, and some more information at the beginning on how the clusters are constructed. And also, if you want more information, I recommend going on the KEGG Pathway database website, where you will have additional descriptions. I hope to see you soon in the Blue-Cloud Hackathon, and I wish you good luck with the using of this notebook. Thank you very much.